I'm just enormously thrilled and honored to be part of this really interesting and terrifying. I didn't know I was going to come and be terrified symposium. But I'll try to be a little less scary in this talk. And what I'm going to do is, there is, along the theme of evolution, certainly it's going to continue in this talk, and I'm going to talk about it in two dimensions, or two levels. One is on the level of what's going on in our population, which poses some very interesting evolutionary questions on why we're having a diabetes epidemic. And then I'm going to talk about some cell biology on a gene that we got very excited about that came from our own search for genes that confer susceptibility to type 2 diabetes. So let me start by talking about a concept that I think many of you are very well acquainted with, which is the concept of a sensitized screen, which can be done which is occurring now in humans, but is more familiar to you in the context of model organisms. The idea is that if you look at the distribution of a phenotype across a population, any kind of population, you see something like this. But if you apply a treatment, usually a stressor of some kind, could be temperature, could be a toxin, could be nutrients, the frequency distribution changes. And the result of that, and the reason it's used in an experimental context, is that it increases the proportion of the population that exceeds a certain interesting threshold, the one that you really want to look for, such as if you are doing a genetic screen for disease susceptibility genes. Right now, the entire world is conducting a sensitized screen. And here's an example of how it's playing out in the United States. These are maps that you can get from the CDC, and they're very dramatic, and they show you just the last 20 years, but it stretches back further, and I'll show you that too, of how this is the percent of the population with a body mass index above 30. So to give you an example, I would have to gain about 55 pounds to get there. And now we're moving from 1994, where very few states were above 15% above 30, to the present time where now we have a lot of red states. And they, okay, so you get the message there. So what that means is that we have a sensitized screen for diabetes, where the sensitizer is diet. The sensitizer is everybody's eating more calories than they did before. And so it's spreading out the distribution, and you have a much higher proportion of people with diabetes. And so sure enough, if you look at the diabetes map, it tracks pretty well with the obesity map. Just to show you what it looks like going back to the 1950s, the left y-axis is percent with diabetes, and it was really low. It's shockingly low by our standards of today. We're down here at less than 1%, and now we're up at around 7.5% of the population, of the US population. But the question is, why? So the heritability of diabetes is about 50%. So about 50% of the variance in diabetes is attributable to genetic factors. And so the question is, why are diabetogenic alleles so common? And one of the things that we see is that it's not uniformly distributed when you look at countries around the world. And these are the countries with the highest prevalence of diabetes. And I'm showing it to you because the ones with the highest, Micronesia and the Marshall Islands, are in the 40s. And it's not just because there's more obesity there, because among obese people in the United States, only about 20% develop diabetes. So that's not the whole explanation. And again, you see a pattern, Pacific Islanders, and then another pattern is the Arabian Peninsula is very high. They're in the mid-20s, which is still extremely high. So one hypothesis that was first articulated in the early 1960s by Neal was that many of these populations have in common a past in which they encountered famine. And that his idea was that famine would select for thrifty genes, genes that would ensure efficient energy storage, and that in the context of overnutrition, these genes would conspire to promote diabetes. Very attractive idea. But there are two problems with it. One is that in famine, people don't actually die of starvation, they die of infection. So it's not clear that the unit of selection is the same thing. And then very recently, now that we have so many genome-wide association studies going on, you can test the prediction of the thrifty gene hypothesis, the prediction being that if you look in the genome, there should be evidence of positive selection for the disease-causing alleles. And that's not borne out. Now, it's hard to interpret negative results. It doesn't disprove the hypothesis, it just means that that prediction has so far not been borne out. And I'm going to leave that hanging a little bit, but it is a very interesting paradox because the fact still persists that these alleles are common, and so it seems like there was some kind of selection, but we don't see evidence for it. Is it because genome-wide association studies are underpowered, or the model of single gene, looking for single gene effects for a complex multigenic disorder is all wrong? That's a possibility. So we just don't know the reason. Now, let me just tell you a little bit about diabetes. So this is what we call a hyperbolic curve. And on one axis, we're looking at the ability to secrete insulin, and on the other is the sensitivity to insulin. And if you're on this curve, that is, if you're matching your demand for insulin with your supply of insulin, you're okay. So you could become very insulin resistant, but if you're that's dialing up this direction, but if you are dialing up your insulin secretion, you'll be okay. Where you become diabetic is where when you dial up, dial down your insulin sensitivity and you're not dialing up your insulin secretion, that gap is what makes you become diabetic. And that's what happens to people who become diabetic. So they're insulin insensitive, but they can't compensate for that. They can't secrete insulin properly. So this means that the low capacity for insulin secretion is pretty common. And again, it poses a question, why is it common? And I'd be curious to know what people think about that, and maybe we can discuss that later. So now I'm going to transition to some cell biology that's behind this, which also has some interesting evolutionary connections. So I'll tell you a little bit about insulin secretion. This is a diagram of a beta cell. Beta cells live inside of the pancreas in little tiny balls that we call islets. So it's a ball of endocrine cells that produce several hormones, but the beta cells are specialized to produce and secrete insulin. And they do so in response to nutrients like glucose, but other nutrients as well. And I like to divide a complicated pathway here into three parts. There's nutrient sensing, there are some signaling pathways, and then there's a bunch of trafficking events that get these little vesicles, these little compartments, these bags, if you like, of insulin, and move them to the plasma membrane where they fuse, and then the insulin is secreted. So these are the things that occur, and you can imagine that along the way there are many places where dysfunction could screw up the entire pathway. And maybe that's a reason why there are so many ways to become diabetic. But it doesn't help explain why these would be common, why there might be positive selection. So now let me tell you about what we've done with mouse genetics. So we've used mouse genetics to look for the genes that make people susceptible to type 2 diabetes. And we were not biased when we went into this. We didn't say, oh, we're going to find something in beta cells. We were very agnostic about where the defects were going to be. This is no longer working, nor is this. Okay, great. Okay, so I always like to show this figure because it makes a very simple point. And I like to call it the obesity-diabetes dichotomy, which states very simply that most people who have type 2 diabetes are obese, but most people who are obese don't develop type 2 diabetes. And it's an important point to frame the genetic problem because we really want to know then, what is it genetically that keeps people in the yellow circle safe and sound in the yellow circle, and what is it that causes, on the other hand, people in the yellow circle to move into that red circle? And that's at the crux of things we've been doing in my lab. And we've done it with a model system in which we have 2 mouse strains that I was going to say personify, that mousify the yellow circle and the red circle. So these guys, it's a strain called C57 Black 6, very widely used strain, which we affectionately call B6, is very obese when we make it genetically obese, but non-diabetic, while these guys, called BTBR, when they become obese, are very diabetic. So in my lab, our goal is to use genetics to do 2 things. One is to identify causal genes that are responsible for the phenotype, what people today call forward genetics, but in the old days it was called genetics. And then the other thing that we do with genetics is to, and in some ways it can be more informative, is not to look for causation directly, but to piece together the pathways in between. And this is where all the omics stuff comes in, and where you can use genetics as an anchor to make models and networks and whatnot, and a lot of fancy math, but it's been very successful. And we've identified lots of both causal and responsive genes over the years and learned a lot about these pathways. So now into some cell biology, and Jim Rothman walked out. This is very relevant to what Jim Rothman discovered and won the Nobel Prize for 6 months ago. And so let me tell you a little bit about it. So this is the inside of a cell, and this is a compartment called the endoplasmic reticulum, which is where proteins are synthesized, and if they're destined to be secreted, they are inside of this compartment, inside of these vesicular compartment, and then they bud into another compartment and fuse with another compartment called the Golgi, named after a man named Camillo Golgi. A lot of people don't know that it's an eponym. And this is a fascinating compartment because it is a place where sorting occurs. It's the postal station, so to speak. Because really, and the metaphor works because there are addresses on proteins that tell them where to go. So a lot of sorting goes on here. And I'm emphasizing 3 types of sorting here. One to the lysosome, which is a digestive compartment of the cell. Another is just straight secretion, constant, and what we call constitutive, because it is constant, as opposed to regulated secretion, which is what we see in the insulin, in the instance of insulin. And so here I'm going to show you that part of it. So with insulin, what we have is a protein that's made as a precursor called proinsulin. It's got an extra piece attached to it and is inactive. And that's true of most hormones that are made. They're made as a precursor. And it is sorted in the Golgi into these specialized vesicles that are very special because they don't release their content unless they're triggered. And in this case, it's triggered by glucose and other nutrients. And so there's something very, very special about these guys as opposed to these guys. And we're really interested in what is so special about that. And again, we don't know. And so genetics is a great way to open up a black box like that and find out. And you know, some of the concepts about sorting have been that there's sorting for entry. Maybe there are receptors, these proteins that might bind to the cargo and say, you go that way. That's one idea. And another idea is that there's something intrinsic about the cargo that allows it to sort itself, not necessarily mutually exclusive. I didn't know anything about this going into it, so I'm talking in reverse. All we did was a genetic screen. And so, you know, we start out dumb. And when we did a genetic screen, we landed on a spot on chromosome 19 that was associated with insulin. That's what you do in genetics. You're just matching up genotypes with phenotypes, but you don't know anything else. And that's the punchline, but it took us years to get to that punchline, that we landed on this particular gene. And we did so, for those of you who are aficionados, and so you can feel my pain, by making 11 subcongenic mouse strains. That is, we're using these tricks to narrow down the position of a gene in a vast genome to a single gene. It took a lot of effort, and I always have to share this because it was difficult. And so we landed on a gene called SORX1. What the hell is that? It's a membrane protein. There it is. It's big. The gene is big, 600 kilobases, a couple of huge exons, and there were some mutations. There they are. So what do we do with that? Well, first of all, we published it, and shortly afterwards, several people found that it is associated with diabetes in humans. That helps. Significant section of your grant proposal. You can always cite that. And you get in trouble if you don't nowadays. But it's also part of a gene family that shares a region called BPS10. And I'll tell you what that is in a moment. But it's an interesting family because there are proteins in this family, like this guy here, sortilin, that are involved in many things. Alzheimer's disease, attention deficit disorder. This thing binds to proteins that cause neuronal death. It sorts glucose transporters. But nothing was known about our gene. That was its cousin. What about this guy? Nothing. But there's one clue, and that is that the ancestor of this is a yeast gene. And yeast teaches a lot, as we now know from Randy Sheckman's work. And this protein is called vacuolar protein sorting 10. But the vacuole in yeast is the same as the lysosome in mammalian cells. It's a sac of enzymes that digest things. It's the cell stomach. And this gene was discovered in a screen by Scott Emmer, a former student of Randy Sheckman, for proteins that are involved in moving from this Golgi compartment, the sorting compartment, to the vacuole. So it's a trafficking protein. And that's all. And that was an interesting clue about its function, that it might be moving proteins to the lysosome. So we knocked it out in mice and made them fat, and they became diabetic. There it is. That was a day of celebration, because you don't know. You just don't know until you do the experiment. Did you get the right gene? Did you make a mistake, or is it nothing? And that's something. So that was very, very nice. And then a very wonderful thing happened to my lab, and that was the arrival of a very gifted scholar named Malcolm Kebede, just a brilliant, extraordinary person, a child prodigy, actually, from Ethiopia by way of Australia, by way of Canada. And right away, she noticed that when she isolated the islets, those endocrine balls that I was telling you about that come out of the pancreas, just under a low-power dissecting scope, you can see that they're different. They're lighter colored. And if you stain them for zinc, and zinc coats the insulin crystal inside of those vesicles, if you stain them, it's obviously different. They're much lighter staining with this zinc stain called dithiazone. There it is. And then if you take a much closer look now under the electron microscope, you see now a very obvious problem. And that is that while these are the wild type, these are normal beta cells at higher mag, And these are those vesicles I've been telling you about. And they have this big, huge crystal of insulin with zinc inside of them. These are called insulin granules or dense core vesicles. And there's about, when you count them, there's about a 75% reduction in these vesicles. So that was pretty amazing. It means that we found a gene that is really important for making these guys. But it's not what you would predict based on what I told you earlier about what happened in yeast. And the neighboring cells that make glucagon are normal. So it's specific for the beta cell, which is sort of interesting. And what happens to the insulin is it goes away. So here's the amount of insulin, just the size of that peak. This is mass spectrometry data. And you see lots of it there, as you expect, and very little of it there. And I won't go into more details, but it looks like it's being degraded, and that's a whole other story. And there's a problem in processing the insulin, so there's an increase in the precursor. Now here's an interesting evolutionary twist to it. It turns out that this gene family, these VPS10 proteins, are also in protozoans. And this is a picture of Toxoplasma. And they have secretory organelles. They have very funny names. They're called rope trees and micronemes. And when they infect or when they're stimulated in a particular way, they don't secrete the contents of these organelles, they secrete the organelles. And they do so in the same exact way as we secrete insulin. They engage the same machinery. Everything's been conserved in evolution between these guys and us. And it turns out, and there was this beautiful paper published 2 years ago, that and a genetic screen, comparable, I guess, to the one we did, identified what genes are needed for doing this, for getting these guys produced, these secretory organelles, and they landed on a gene that's in exactly the same gene family as what I'm talking about, another VPS10 gene in protozoans responsible for these micronemes and rope trees. And there it is. So this is the wild type. And you see these are the rope trees here, and these little guys are the micronemes. And if you search this picture here, you don't find them. This is the knockout. Pretty similar phenotype. And then just this year, Aaron Turkowitz at the University of Chicago found a very similar thing in Tetrahymena. There they make these things called mucocysts, and they also have a defect in mucocyst formation when you knock out their VPS10 gene. So what that means is that in evolution, we went from a protein that in yeast was moving things from this sorting compartment to the cell stomach, the lysosome, or in yeast it's called the vacuole, to protozoans where a cousin of this protein changed its function to making secretory organelles. So we looked at it more closely and asked, well, where is it in the cell? And now you won't be surprised in light of everything I told you, but it is beautifully colocalized with a marker of the Golgi. And this is the nucleus of the cell, and this location, is called perinuclear. That's what we always say when we see that. So it's a very nice localization at the Golgi. So we have a simple idea that this part of the molecule is sitting inside of that vesicle, and it binds cargo. And then there's a membrane, and then this part is extending into the cytoplasm and is responsible for the postage stamp and the address, and is moving it around the cell. So one idea that we had then was that if we deleted this part of the molecule, the cytoplasmic piece and the part that anchors it in the membrane, and just left it with this domain, which we call the luminal domain, so we call it Lum-Sorcs 1, that it would bind the cargo but not be able to move it properly, what we call a dominant negative. So it's going to behave as if it's like the knockout in the animal. We wanted to create a model system in cells. It's easier to work with cells. And it worked. And so here it is. This is the insulin in the wild-type cell line, a beta cell line, and this is the insulin in the cells expressing this dominant negative. So it worked. It's binding cargo, and it's got nowhere to go, and so the insulin is getting degraded. And these are the vesicles here in the wild-type cells, and these are the ones in the ones expressing the dominant negative. The vesicles go away. So it's duplicating exactly what we saw in the knockout, and here's the quantitation of that. Here is the location in the cell of insulin, and you see it widely distributed because it's in all these granules all over the cell, but then when you knock out this guy, it's stuck. It's stuck in the Golgi. It has nowhere to go. So I'm going to show you another fun experiment, partly because it's just the technology of cell biology is so wonderful that it's fun to just talk about these things in their own right, and of course the result is interesting. But this is a very nice probe that we didn't develop. It was developed by somebody else, which is a red dye, basically. It's a protein that folds and forms and has a color. And it's called DS red, but it's not red initially, it's green. So it starts out green, and then 30 hours later it turns red. And so it's a timer. So it's a timer in a cell. And we're really interested in this because it turns out that these insulin vesicles are discriminated by age. There's age discrimination in the cells. The younger ones are preferred. So when the cell decides which vesicles to choose to secrete, it chooses the younger ones. And so we decided that we would label the younger ones or look at them selectively by looking at only the green ones, by putting a marker inside of them with a protein called syncolin. And the question we were asking is there a defect in replenishment, or is there a turnover of these granules? Because I haven't told you why they go away. They could be because they're not being made or because they're being turned over. And this is a way to ask that question. And so what we did is we labeled them, and here they are. They're all green, and they're everywhere. And then we exposed the cells to high glucose, which triggers secretion. And I told you the young guys are favored. So you see a depletion because they have been secreted. So there's a depletion, and then there's replenishment after that. But in the ones expressing this dominant negative, you see depletion, but now you see this accumulation at this pole, like I showed you before, the Golgi, and there's no replenishment. So using this timer, we're able to tell that there's a defect in making these guys. And so what we're seeing then is that this protein, we're placing it here. This is our model. There's a lot more to do, obviously, but we're saying that it is functioning at a step after the Golgi, after proinsulin is converted to insulin, and is necessary for pulling it away into these regulated vesicles. And in the absence of that, the default is to transport it to the lysosome where it's degraded. And finally, and this might have some therapeutic significance if we could figure out a way to agonize this pathway. If we go in the opposite direction and we ask, I showed you that it's necessary for making granules. Is it sufficient? That's a much higher bar to rise to. And the answer is yes. So if we overexpress, we put it on an inducible promoter. So in the same cells, we induce the expression of this gene. We go from this many vesicles to this many vesicles. And that's the quantitation. It's a pretty good increase. So I've told you two evolutionary stories. One of them has to do with selection at the population level. And the other has to do with the appropriation, the tinkering. Evolution is a Rube Goldberg tinkerer, actually. It works with what it has, and what it had was a very good cargo transporting, trafficking protein, and it adapted it to go from moving proteins to the lysosome to manufacturing and transporting proteins to secretory organelles, first in protozoans for these secretory organelles, and then in beta cells for insulin-containing granules. These are the people who've done the work, this magnificent scholar and a technician with whom she partnered. I call them the dream team, and various collaborators. Thank you.
Okay, we have some time for questions.
How often do you find SORCS1 mutations in diabetic patients? Nobody's looked.
These are, the genome-wide association studies are extremely insensitive because the number of markers or SNPs that are interrogated is in the millions. And so there's a statistical penalty for doing that. And so the false negative rate is extremely high. So to answer that question, you have to do something much more expensive. You have to go and sequence the gene in lots of people, and nobody has done that. Larry.
Stay here. Sit down. So this is an earlier question. This notion that there are thin people who get type 2 diabetes, 20% of the diabetics are thin. And do people study diabetes in thin people and try to understand if there's a difference in those populations?
Yes, absolutely. And so that's actually more common in Asia and Africa. I think where we are with diabetes is where people were with cancer 50 years ago, where we don't have a lot of nuance. We're calling many things type 2, but it really should be type 2 through 50. We need a more molecular definition. My hunch is that, as you saw the pathway in the beta cells, there's so many ways to screw it up, and so there are going to be defects in different steps of the ones that I outlined. There might be defects also in the ability to proliferate those cells. And that might lead to a better, more nuanced classification system. Yeah.
Thank you. It was very, very wonderful lecture. Just a clarification, I had always thought that type 2 diabetes, when you called it insulin resistance, was more of a receptor problem than an insulin production problem. Is this when you're saying there's going to be many, many varieties of type 2, or is that an erroneous assumption?
So that used to be the prevailing view 10, 15 years ago, because most people who are obese are insulin resistant. But that has changed for two reasons. One is that a lot of the genes that are involved in sensing insulin and transmitting the insulin signal have been knocked out, creating extremely insulin-resistant transgenic mice, and they're never diabetic. So in other words, insulin resistance by itself is just not sufficient to cause diabetes. That's the biochemical evidence. The genetic evidence is that virtually all the genes that have been found in human populations that are susceptibility genes for diabetes play out in the beta cells. So type 2 diabetes is almost entirely a beta cell disease. You can think of insulin resistance as the sensitizer. It brings it out. Because all these people who had limited beta cells had them in 1950 and didn't have diabetes.
Hi, thank you very much. I just have a question. What about epigenetic factors with diabetes? And we have this tremendous growing population, wide diversity, changing in eating habits, changing in environment. What kind of work has been done around the epigenetic
That's a great
factors?
question. So the first evidence we ever had so the question is about epigenetics, and epi usually means on top of genetics. It means in addition to or factors that, and what we really mean it to be, is how the environment can cause heritable changes in people. It's validating Lamarck in a way. You could call it that. And the first evidence that that could be relevant for diabetes actually came from studying children of mothers who endured the Dutch famine in the end of World War II. And they studied them very carefully because there was a high incidence of diabetes in those children and in their children. They've been followed for several generations. And it had to do with a different, what particular stage in gestation the mothers were in when the famine hit. So that was a very inspiring and very important study and motivated a lot of animal work where the same phenomenon has been reproduced in experimental animals. And now there's a lot of work going on asking what particular nutrients and what role might those nutrients play in modifying the DNA itself or in modifying proteins that bind to DNA and change gene expression, and it is a very, very active area. I'll repeat the question.
So I want to link into the previous talk on the abusive use of antibiotics, and wanted to know your view on the role of the microbiome in developing those metabolic diseases.
Well, I'll address it more to the obesity side of things than the diabetes side because we don't know as much on the diabetes side. But it's hard to believe that microbiome isn't going to be relevant there. I'm almost certain it is, and we're doing some work on that in my lab. On the obesity side, there's some really extraordinary work from Jeff Gordon's lab at Washington University, where he has taken the microbiome organisms from obese humans and lean humans and transplanted them into what are called gnotobiotic mice, mice that have been raised under sterile conditions and have no microbiome. So they have only what he gives them. And the microbiome from obese humans make the mice obese. So that work is being extended to try to ask what small molecules are being made by these bugs that regulate, have maybe endocrine effects in people, and what do they do in the intestine itself, where there's nutrient absorption, and there are some very important neuroendocrine cells that talk to the brain and affect food intake. It's very active and it's a game-changing area completely. And I noticed Jeremy Nicholson is here, and he's a leading person in that field.